Hello guys, this is the second part of the development of the nervous system. In previous lecture we have discussed the formation of neural tube, and in this lecture we will proceed forward and we will talk about the formation of brain and spinal cord from neural tube. So we will talk about the development of nervous system. Development of nervous system. So first, what happens after the formation of neural tube? Neural tube, three primary vesicle appears. Three primary vesicles appears. So what happens? These vesicles are proencephalon, mesencephalon, and rhombencephalon. Then what happens? These three primary vesicles differentiate into five secondary vesicles. The proencephalon is further differentiate into telencephalon, and diencephalon. And thank God, mesencephalon remained the same. And rhombencephalon is further differentiated into metencephalon and myelencephalon. Metencephalon and myelencephalon. So after that, what really happens? Telencephalon is the future cerebrum, and diencephalon gave us thalamus and its related structures. And mesencephalon will develop the midbrain, and metencephalon will further differentiate into pons and cerebellum, and myelencephalon will develop medulla oblongata. So this was a very short review on the development of central nervous system or brain and spinal cord. So now we will try to understand these structure diagrammatically. So now, as we discussed in previous lecture, the central nervous system appears at the beginning of the third week as a slipper-shaped plate of thickened ectoderm, which is known as the neural plate. In the mid-dorsal region in front of the primitive node, its lateral edges elevate and forms the neural tube. With further development, the neural folds continue to elevate and approach each other in the midline and finally fuses, forming the neural tube. Fusion began in the cervical region and proceed in cephalic and caudal directions. Once fusion initiates, then it forms the two pores, the anterior neuropores and posterior neuropore. You can see these neuropore in this diagram like this: anterior neuropore and posterior neuropore. So one thing which is very important to remember, the anterior neuropore is closed at 25th day and the posterior neuropore closes 3 days later, or day 28. Then what happens after closure of these pores? The neural port you will see like this, and of course it has a central C-shaped filled cavity, Then what really happens, three bulging or three dilations start developing in the neural tube. So these dilations are known as primary vesicles. So in simple, primary vesicle start developing at the cephalic end of neural tube. By end of 5 weeks, these three primary vesicles differentiate into 5 secondary vesicles. So these primary vesicles, as we discussed earlier, that this is known as the proencephalon, mesencephalon, and rhombencephalon. By the end of 5 weeks, these three primary vesicles differentiate into 5 secondary vesicles. The mesencephalon remain the same, and the rhombencephalon will further differentiate into metencephalon and myelencephalon. So this one is the proencephalon will differentiate into telencephalon and diencephalon. And then, thank God, mesencephalon remain the same. And rhombencephalon, it will differentiate into metencephalon and myelencephalon. So that what really happens, from telencephalon, the two cerebral hemispheres will develop, and from diencephalon, the thalamus, hypothalamus, pituitary, and optic vesicle will form, and from mesencephalon, of course, the midbrain, and from metencephalon, pons and cerebellum will form, and from myelencephalon, medulla oblongata will develop. So of course there is the central CSF field cavity. This is the central CSF field cavity, and it will diverge laterally along with the brain, along with the hemispheres. So the lumen of the spinal cord, the central canal, is continuous with that of the brain vesicles. So the cavity of the rhombencephalon is the fourth ventricle. That of the diencephalon is the third ventricle, and those of the cerebral hemispheres are the lateral vesicle. You can see like this. And the lumen of the mesencephalon connects the third and fourth ventricle, and this becomes very narrow and is then known as the aqueduct of Sylvius. Each lateral vesicle communicates with the third ventricle through the interventricular foramen or foramen of Monro. So now, after the formation of 5 secondary vesicles, that is telencephalon, diencephalon, mesencephalon, metencephalon, and myelencephalon. So this is the anterior view. If we draw the structure of the brain posteriorly, you will see something like this. This is the telencephalon, and this is diencephalon, mesencephalon, metencephalon, and of course myelencephalon, and then the spinal cord. What really happens at metencephalon, bulging will start growing like this posteriorly, and it will develop the cerebellum. So posteriorly, this is cerebellum, and anteriorly you can see here this is pons. So what really happens, if we see the structure of the brain from side view, you will see something like this. This is the telencephalon, and a small structure is diencephalon. Then we can see this is the mesencephalon and metencephalon and myelencephalon, and then spinal cord. So it will form a structure like this. And of course here is this is the cerebellum. And then what will happens, the telencephalon will grow further. Then it will form the frontal lobe, parietal lobe, occipital lobe, and then this is the temporal lobe. So now this is the structure of brain. So this one is called the two cerebral hemispheres, and then the diencephalon, the thalamus is present here, the midbrain, then the pons and medulla oblongata, and then cerebellum. So it was all about the formation of the central nervous system. So now we will discuss the formation of spinal cord. Then what happens if we take a section from this part of the spinal cord? You will see something like this. So this is the central canal, and around it these are the fibroblast cells. So then what happens? These fibroblast cells will start growing posterolaterally, and it will make the alar plate. And then some cells will start growing up anteriorly, and it will make the basal plate. So the alar plates, these are concerned with the sensory function, and the basal plate concerned with the motor function. So this is the anterior fissure. This is called the anterior fissure. And then this is the posterior sulcus. So it was all about the development of the nervous system, and I hope you have understood, and thank you for staying with us.